All right. Well, first and foremost, I hope everyone is staying safe and sound. Hopefully the next time we can have this meeting in person. I would like to thank everyone who has worked really hard at EMSL and PNL to make this meeting possible. I also would like to thank Ying Zhu for giving this opportunity, and as well as the other organizers, to highlight some of the technological developments that my laboratory has been doing to enable single cell mass spectrometry in actual live embryos. So we've already heard about the importance of cell heterogeneity, and Ryan beautifully highlighted this field and the challenges. So I will just focus on the main topic, at least for this conversation, and that is really that individual cells are critically important in development. We have a vast amount of molecular information now that demonstrates that two cells are not created equal. These cell-to-cell differences are implicated in disease, in normal development, and that's really what I'm going to focus on today, normal development, specifically in regards to embryonic development, where in space and time, all the different kinds of cells must be established from a common ancestor cell, that is the fertilized zygote. But of course, these developmental processes do not stop at the development of the embryonic body, because we also need a brain, obviously, and there are hundreds of billions of neurons that all do something different, and how wonderful it would be if we had the technical tool set to understand molecularly speaking how those individual neurons differ from each other. So the challenge has been that most of our technologies measure large numbers of cells, however, unfortunately, at a level of an entire population by averaging together the signal. And this average signal that I'm highlighting here does not necessarily highlight what the underlying populations of cells would present in terms of their phenotypes. To understand cell-to-cell differences, we'd like to take a molecular systems cell biology approach by integrating genomic, transcriptomic, proteomic, and metabolomic information. So we do have really good tools, thanks to next generation sequencing, to study single cells and single neurons in high throughput, including, for example, the differentiation of single cells as they form mature neurons from neuronal progenitor cells. But we don't really have these tool sets for the proteome, and that's really where my laboratory comes in. How do we develop new generational mass spectrometry tools to enable the characterization of hopefully large numbers of proteins? So Ryan, Kelly, and my laboratory has been tackling this challenge for some time now. Now, before I show some of our tricks, I would just like to welcome you to our laboratory to see what types of embryos we're dealing with. So in this figure, what you will see in this movie, what you will see is how a single cell, these are individual embryos at the one cell stage, quickly develop to give rise to multi-cell organisms. 2 cells, now you see 4 cells, now you see 8 cells, now 16, 32 cells, 64 cells, 128 cells, and so on and so on. Each of these divisions happen in about 15 cells, 15 minutes. So that means each of these single cells only exists for 15 minutes in our laboratory. That is all the time we have available to perform our single cell mass spectrometry measurements. That is quite a difficult challenge in terms of analytical chemistry. Now, one would think that we can just simply measure transcripts using single cell transcriptomics, but the inconvenient truth is that transcriptomics is not the same as proteomics. Mark Kirshner's and Steve Giggy's work recently in developmental cell is one of my favorite. I often mention, because they correlated the actual expression of proteins with mRNA, and what they found was a large number of proteins that very nicely track each other in terms of gene expression at the level of transcription and translation. But we have really interesting cases where gene expression, so the production of transcripts, does not correlate with the production of proteins. There can be transcripts gone by the time the actual proteins are made. And there are very nice, insightful comments on how this can happen in this publication. So the idea here is that we need to measure proteins if we would like to complement single cell transcriptomics. But how do we do this in a developing embryo? And so here I just slowed down development by taking these snapshots, and I'm quickly going to go to the 16-cell embryo because that is primarily what my lab focuses on. So in the 16-cell embryo, we can actually identify particular cell types in the frog, the South African clawed frog called Xenopus laevis that my lab actually uses as biological model to stem cell differentiation and development. And so we know that in this model, this particular cell, the D11 cell, is reproducibly precursor to the brain, the retina, and the spinal column of the frog, so neural tissues. In contrast, the V11 cell, highlighted here, is precursor to the epidermis. Now these cells, although they seem gigantic, 250 microns in size, they only contain 990 nanoliters in volume. And so that is already a challenge, not to mention that these cells are not occupying a flat array, but instead they are populating a truly three-dimensional object, the embryo itself. And so how do you access these cells is now the question. So my lab has developed various tools to address this, and one of the first developments was to develop an ultra-sensitive, small volume analysis system to measure proteins based on capillary electrophoresis. So what do we do? So what we designed was this platform where we're able to manually deposit about 200 nanoliters of sample into this sample loading microvial. So this may be viewed as almost like a precursor of the microwell plate that Ryan and Ying have beautifully automated into nanopods. Now what we're doing differently, obviously, is that we're using a capillary that we position over to inject 10 nanoliters of sample into this capillary, and then we apply about 20 to 30,000 volts across this capillary so that we can electrophoretically separate out the biomolecules, in this case, the peptides in this capillary. Now, the story does not end there because obviously we need to ionize these biomolecules. And so how do you do that with CEE is also not a trivial task. So piggybacking on Dick Smith's pioneering work from the 80s, we actually custom built a coaxial sheath flow interface, a CEESI interface. We've made a couple of advances, including the active control of the spraying regime, so that we can efficiently ionize the biomolecules, the peptides, for identification via sequencing and then quantification by label-free and label-based approaches. Now, just as Ryan highlighted, every single step is important when you do a single cell proteomics. So just as Ryan, I'm also highlighting microsampling. How do you aspirate the sample? How do you separate? How do you ionize? And how do you detect this in interconnected steps, with each of these steps being equally as important? This is just to give you a bird's eye view of a corner of my lab. Here you can see the actual commercial mass spectrometer. And then this is the platform that we have custom built together with commercial microscopy, so that we can identify the cells, extract, process the materials, present the material in these very small amounts for electrophoresis, and then use Orbitrap tandem mass spectrometry for identification and quantification of proteins. Now, just as any good analytical chemistry experiment, you first need to validate that your platform is reproducible, it's robust, it's got the right figures of merits, and we're no exception to this rule. So that's what we are looking at now. So we took model peptides to simply validate the linear dynamic range of quantification and a lower limit of detection for our platform. We have about 1 to 3 log order demonstrated here, but we actually have about 5 to 6 log order of linear dynamic range of quantification. Our limits of detection are about 25 attomoles back a couple of years ago. We can quantify proteins in a label-free approach with about 10% relative standard deviation in terms of error. And one interesting aspect of doing cap electrophoresis over liquid chromatography for general proteomics, not just for single cell analysis, is the very different mechanism by which separation occurs. Now, here you can see these nice trends based on size and charge in CE, which obviously we don't have in liquid chromatography. I'd like to draw your attention, though, to the sensitivity improvement that micro CE in our hands offered compared to nano liquid chromatography. From 200 times smaller amounts of starting material, we were able to identify comparable, if not maybe a little bit higher number of peptides by CE versus LC. And there's many fundamental reasons for this, which I will not have time to explain, but you're more than welcome to follow our publication track record where we discuss the basics of capillary electrophoresis and how that advances sensitivity for bottom-up proteomics, specifically for single-cell proteomics. So over the years, we have developed proteomics and also metabolomics workflows to measure proteins and metabolites in actual single cells, small amounts of tissues, small populations of cells in Xenopus laevis, zebrafish, mouse, you name it. And without going too much into the details, I'll just quickly go to quantification. Now, we very quickly started asking biological questions in this workflow. For example, how do we even know whether these cells, the D11 and the V11 cells in the embryo, which give rise to, reproducibly give rise to different types of tissue fates, whether they contain different types of proteins? Now, the level of transcription, there is some evidence of cell-to-cell differences, but these transcriptomic differences are actually lost between many of these cell types. So that's what we started mapping out for developmental biology. How do we do this? Well, in this model, we are able to dissect out the cells because these cells are really large. Then we did a bottom-up proteomic workflow starting in small volumes down to about 1 to 5 microliters of material in classical vials. And then we tagged the resulting peptides with tandem mass tags so that we can now pull together the bottom-up protein digest from actually three different cell types, and those are D11, the V11, and the V21. So neural tissue faded, epidermally faded, and hindgut faded cells. And now we've got this 1 to 5 microliter of sample that we're going to measure in a bottom-up proteomic approach using our custom-built micro capillary electrophoresis, nano or microflow electrospray ionization, CE nano electrospray ionization ion source, followed by various kinds of Orbitrap tandem mass spectrometry approaches. An important point to note here is that we're not actually measuring the entire cell's protein content in our workflow. We're only measuring about 10 to 20 nanogram content, which is about 0.2% of the total protein content contained in these blastomeres in these single cells. And what that allowed us to do actually is to identify almost 2,000 protein groups already about 4 years ago. And we were actually able to identify these proteins in a cell type specific manner. So that's what you can see here, how the identified proteins are matching or not matching between the V11, the V21, and the D11 cell types. Now, without going too much into the underlying biology, what I'd like to though highlight is that these differential identifications, these cell type specific identifications actually make good sense, it turns out, because you were able to find proteins that are known to either be ubiquitous in these cell types or proteins that are uniquely expressed in particular cell types, such as the V21 cell, which is the future fate of the Proteome, or for example, the D11 cell, which eventually then gives rise, which contains gemin and isthmin, which are known proteins with significant roles in neuronal development, which is the actual fate of the D11 cell. So these differential identifications make sense. We can also relatively quantify the proteins between the cell types using tandem mass tags. And in this volcano plot, correlating the significant differences to fold changes, we can now identify all these proteins out here and all these proteins out here, which are significantly dysregulated at the level of statistics and also fold change between these three cell types. And now that all these proteins are actual fair game for functional experiments, do they have any kind of developmental significance? So this is where now we can generate new hypotheses to drive cell differentiation and molecular understanding of neuronal development forward. And what's really cool, I think, is to correlate the single cell proteomics data to actual single cell transcriptomics data, which we have done a good couple of years ago already. And we were in this experiment, we manually correlated our single cell proteomics data to about 900 different genes that have been detected also at the level of single cell transcriptomics. What's really interesting in the interest of time is that we were able to actually identify dorsal, ventral, and animal vegetal differences between individual cells that occupy these primary developmental axes, which were not detectable at the level of transcription. So this really piggybacks on the earlier point I was making. mRNAs are not proteins. If we would like to understand the system cell biology view of cell differentiation, there's no way around. We got to measure the proteins as much as the transcripts. And of course, we can't stop just there. We also need to measure the metabolites as well. Now, of course, the question that we got very early is how do we handle smaller cells? As the embryo develops, very quickly the cell size becomes smaller, 100 microns, and then very quickly that, of course, becomes 10, 5 micron size, just as a regular somatic cell. How are we going to measure proteins in these small cells? So obviously, manual dissection is not the way to go. And we developed a microprobe sampling approach where we used precision fabricated capillaries, somewhat similarly to what Thomas Brown highlighted, albeit for a different application. In this case, we took a capillary that we connected to a microaspirator device so that we could identify actual particular cell types in live embryos. So in this case, I know exactly this is a dorsal faded cell in the live embryo. Then we used multi-axis translation stages to position the capillary into the identified cell. Then we coordinated a pressure pulse to aspirate the content of the calibrated portion of the cell in, so that we can aspirate it into this capillary. That is the white material you can see here. This is the actual cytoplasm collected from the cell, and then we could expel the material, do bottom-up proteomics for bottom-up proteomic identification and quantification using our custom-built approach. What is cool with this approach that I'd like to draw your attention to is that compared to microsampling by dissection, this approach is much faster. Instead of 5 minutes per cell by dissection, we can actually collect the sample in just 5 seconds. Further, once you sample an actual cell, the damage on the membrane heals, and that cell actually goes on to develop what appears to be normal. So here you can actually see that sampled cell twice, which then continued to undergo live embryonic development, division specifically. We have even raised more recently up tadpoles and larvae from these microsampled embryos. So you might view this as the first example of truly in vivo single cell mass spectrometry, where actual proteins can be identified in combination with functional experiments. Now, why am I so excited about capillary microsampling, you might wonder? Well, it's because this approach is scalable in space and time. So if this is an actual D11 cell, which it is, that then quickly will divide to give rise to entire cell clones. These 2 cells here, which then divide to give rise to about these 4 cells, and then those develop to give rise to about these clones. All these cells are eventually on the path to becoming neural faded. So what we started mapping out directly in the live actual vertebrate embryo is how the single cell proteome is reorganized as this identified cell becomes forms the offspring, the progenitor cells. And what we found was that the actual proteome in these identified cells in a 16, 32, 64, and 128 cell Xenopus laevis live embryo is indicative of the actual cell type. Furthermore, we're able to, using relative quantification, to also find proteomic changes as the actual cell clone develops. So we have proteins that, for whatever reason, are up regulated in expression. There are proteins that are down regulated in expression, and these are probably the maternally deposited proteins. Xenopus is known to have proteins that are deposited by the mother, and those proteins progressively degrade until the actual cell will make its own protein, Speaking of which, we also detected these transient signals essentially for proteins in the embryo, which coincide with important developmental events such as zygotic transcription and mid-blastular transition. And so these transient profiles are not a fluke in the measurements. They actually line up really nicely to biologically important processes. We are also able to identify transcription factors in these measurements, which highlights the sensitivity of the platform, including even proteins that are associated with the Wnt-BMP pathways. Now, we didn't just quite stop there. Obviously, we would like to go to smaller and smaller cells, including the final somatic cells from these embryonic stem cells, as well as single neurons in mammalian systems. So we recently developed this new generation CE nano ESI ion source, where we use a taper tip emitter that allowed us to bring the emitter source much closer to the mass spectrometer to better entrain the droplets and also to facilitate ionization by avoiding dilution factors. We actually improved the sensitivity by about 10-20-fold, which allowed us to now obtain about 210 zectomoles of sensitivity for model peptides, or 126,000 copies. In the interest of time, I would just mention that using this approach, we can actually measure picograms of protein digest from model systems such as bovine serum albumin and cytochrome C. And more recently, we were able to apply this technology for protein digest from mouse neurons, primary neurons, allowing us to identify about 700 different protein groups from just 500 picogram of protein digest, including neuronal markers that are used in neuroscience and various types of proteins. Obviously, lower abundance proteins are now the next mission by us and also by Ryan, I'm very sure. Now, in conclusions, the single cell mass spectrometry can also be done using custom built tools such as capillaries, home built CE instruments, new generation mass spectrometers. This approach we have developed in our lab is quantitative, and it has allowed us and also other groups to ask new kinds of questions in cell and developmental biology and neuroscience. I'm excited because this now promotes new questions in basic biological chemistry, basic cell and neurodevelopmental biology, and also health research. Of course, this work would not have been possible without the really hard effort of all the students who are doing this work, and together with collaborators, Sally Moody and Chiara Monzini, as well as all the funding agencies who have made this work possible. Thank you for your attention. If you have any questions, I could take them potentially now, or please feel free to submit them via the Discord channel or emails if that works better. Thank you again for your attention.